Okay, can everyone see that?
Yes.
Alrighty. Okay, so here we go. For those of you who know me, it won't be any surprise that this is focused on fungi, on mushrooms, the things that I pay attention to most of the time. But for those of you who don't know me, I want to just give you a little introduction to the organisms that have captivated me to the point that I haven't really gotten away from them, except recently when I've started branching back out and paying attention to other organisms. But fundamentally, it's fungi that have my heart and my attention, so I want to introduce each of them before I get into this anymore. Most of you are familiar with something like this, a big polypore mushroom that grows on decaying wood in the forest. These are familiar even to people who don't know anything about mushrooms, and they're really important because they're wood decay fungi, and this is what we learn, almost all of us in middle school and high school, about fungi. They're decomposers, they recycle nutrients, and in this case, they're recycling carbon dioxide back into the atmosphere and keeping the great chain of plant life going on, as well as your and my life. But this isn't the only thing that mushrooms do. They're also parasites. This is Noah's photo of Cordyceps militaris, I think from Humboldt.
Yes,
Yeah, Joanne, maybe you were there for that one.
I was.
You were. Lucky
Yes.
you. So this is a really rare mushroom in California, but it's a pretty common group elsewhere in the world, especially in the tropics. And this is a famous group for being parasitic. They do all sorts of interesting things to their insect hosts. Some of them control their minds, and I mean that literally. It's actual, like, honest to God mind control through mechanisms that we're not really familiar with yet. It's pretty complicated, and we're just starting to study it in detail. But this is another thing that mushrooms do. These aren't recyclers. These are actually going out and killing or harming another organism, which we tend to think of as negative. But there's extremely important ecological roles that parasites play in balancing ecosystems, in making sure that organisms are at a sustainable density, that they are consuming resources at a rate and at a concentration that can be sustained by the environment they're living in. So parasitic fungi are also really important. And my favorite group probably is the group exemplified here by Chantrell's. These are the mycorrhizal fungi, and they are symbiotes, but so are the parasites. These are symbiotes in a different way. These are mutualists, and mutualist fungi are those that have a positive effect on their host, and the host in turn has a reciprocal positive effect on their fitness. So the case with ectomycorrhizal fungi is that they use the extraordinarily high surface area of their mycelium, which is underground, out of sight, to gather nutrients, to gather water, and to send it to a plant host that produces sugar and sends it back to them. So there's a two-way flow of resources going on here. Soil resources are mobilized and aggregated by the fungus and sent to something like an oak tree or a pine tree, and in turn, the sugar that that tree has made out of carbon dioxide and water and sunlight is sent back to the fungus, and they sort of live together in this mutual exchange of resources for sometimes decades, sometimes longer than that. 100 years is probably not uncommon. So this is another way that fungi shape terrestrial ecosystems. They are sort of at the base of all life on land, because they were probably there when algae colonized land, when they moved out of water and started taking over terrestrial habitats. So this is an extremely ancient relationship that has only gotten more complex and more co-evolved over time. And I really enjoy, especially this group, studying the coevolutionary ecology stories that emerge from it, because there's many very specific fungi to their hosts. So they may only partner with one or two hosts and no others, whereas some of the other ectomycorrhizal fungi are generalists, and they can form associations with a wide variety of trees over the world. And they're really adaptable in that way, which is fascinating in its own right. Another reason that I really love fungi is that they span scales, both in time and in space. So here's an example of a mushroom, believe it or not. I don't know if that is recognizable to anyone, but that is a Copernopsis. It's a little inky cap that grows out of elk dung, and it's minuscule. It's minute. That is probably as large as my pinky finger. Well, as tall as my pinky finger. It's actually substantially smaller than that. It weighs almost nothing. It probably fruits and grows up and matures and releases its spores and then wilts away in the matter of 24 hours or less. And its entire mycelium is restricted to a little pellet of elk dung, which is a pretty humble abode, but it has its role there to gather up all the remaining nutrients that are in that pellet of elk dung and release them back to the environment. So this is an example of a really tiny mushroom. But it gets even smaller. These are yeast cells, and yeast are fungi. This is Saccharomyces, something like the yeast that ferments your wine or your beer or your bread. An extremely important organism from a human perspective. And their life cycles might be hours long or a day long. And they're microscopic. So all the way down to microscopic organisms. They never form a macroscopic fruiting body the way a mushroom does. But this is the small end and the short-lived end of the fungal spectrum. There's also enormous fungi, like these honey mushrooms. And you might think to yourself, looking at these, that they are not particularly enormous. They're just mushrooms. But the mycelium, the underground part, the part that you can't see, is in some cases 2,000 or 3,000 years old, and in some cases spans a landscape scale. So entire mountain ranges may be covered by something like a single individual that has a lifespan measured in millennia. So these are some of the largest and oldest living organisms on Earth. So now we see that the fungal kingdom has these two extremes in it: extremely small, extremely short-lived, and extremely large, extremely ancient individuals. And this is the kind of thing that really fascinates me about fungi. They're not like plants in this way. They're not really like animals in this way. They span the extremes of temporal and spatial scales in a way that I think is really unique. It makes them very mysterious to me. And just to show you just a few of the sort of extreme habitats that fungi inhabit that I find really compelling, really beautiful, really evocative. This is a fungus that was described recently from the high desert of the Andes, where there's extensive salt flats, dry lake beds. And you can see in the foreground of this picture, there's just low mounds of vegetation and big crusts of salt everywhere. And that row of orange dots is actual mushroom caps. Those are a fungus that grows in this extraordinary dry habitat at extremely high elevation. You can see the snow-capped peaks in the background. Here's a closer view of them. This is Lichenomphalia, I believe it's Andina, growing out of this salt crust. So an extremely difficult environment to live in for any organism, but fungi in general are not very salt tolerant usually. So it's an extraordinary thing to think that you can go up to the Bolivian Altiplano and be on these dry lake beds where there's salt crust everywhere, and potentially even flocks of flamingos flying under snow-capped peaks, and there will still be mushrooms there dotting that salt crust. If I die tragically, I want my ashes to be spread somewhere like this, just in case you all know now. One of you can do it. But there are other extreme environments that you wouldn't expect to find fungi in that you can. These are not depicting the same fungus here. So at the top, there's a scanning electron micrograph of a fungus called Xeromyces. And it's also a salt lover. It was found first on the crust of salt on Portuguese salt cod. So this is an ancient method of preserving fish, where you just take a big fillet of cod and you crust it in salt and pack it really tightly on top of each other until you basically have made stiff pieces of wood out of fish, and you have to soak them for days to even be able to eat them. But in that salt crust that preserves the fish, this fungus called Xeromyces can be found. And one of the most amazing things about it is that it appears to have the lowest requirement for water of any living thing. So we often think of mushrooms or fungi in general as being organisms that live in damp, dark, wet places, but that's not always the case. And this fungus is sort of the most extreme example of that. It needs water so little that if you give it extra water, it dies. It really likes extreme osmotic stress, and that's where it thrives. Down at the bottom is a picture of a hydrothermal vent, which I have not yet been lucky enough to visit myself. It's sort of a dream of mine, but I don't know how it would ever happen. But if you take a submersible all the way down 6,000 feet deep or more and find one of these fumaroles, is what they're called. They're areas where water has infiltrated the crust of the earth and is getting heated up down in the upper layers of the mantle, or probably not that far, but it's getting hot anyways as it sinks down towards the core of the earth and gathers nutrients from the bedrock or minerals from the bedrock, and then spews back up into the ocean in these really dark clouds, these black smokers. If you take a little bit of water from around these hydrothermal vents and plate it out on a petri dish in the laboratory, apparently you'll find fungi. They're down there doing something that we don't know. We don't know what the fungi look like. We don't know what their names are. We don't know what their ecological roles are, but they are in deep ocean ecosystems. just like they are on land, just like they are in the high atmosphere, little spores floating around in the stratosphere. So I've told you now that fungi have 3 different main ecological roles, much more ecologically diverse than things like plants and animals. They span the spectrum of time and space, from being really small and really short-lived to being really enormous and really ancient. And they're in sort of the 3 main layers of our experience of planet Earth. They're in the air, they're on the land, and they're in the water, even down to the ocean's depths. And these are the reasons, among others, that I am fascinated by fungi. They contain multitudes in the truest sense. But fungi are also found in you and in me and on you. This is a picture of my brain, and thankfully it doesn't show any signs of fungal colonization, but that isn't a fact that we can take for granted. Recently, fungi have been found to live in the brains of patients with Alzheimer's. And whether or not that is a causal relationship, we don't know yet. We don't know if they simply opportunistically colonize the brains of patients that are already experiencing a disease, or if they somehow have a role in causing that symptomology. But this is sort of a morbid example, but there are also positive examples. Fungi play a major role in our microbiomes, in our guts and on our skin, in ways that keep us healthy and prevent us from getting other infections and make our lives better. You know, the microbiome in our guts allows us to absorb nutrients more efficiently and to synthesize neurotransmitters in ways that we're just beginning to understand, and I'm really fascinated by. Eugenia Bone wrote a book called Microbiome. I think that's, or Microbia, I forget what it's called, but it is on my to-do list to read. I think Ed Yong also wrote a book about this. He's a New York Times science columnist who always writes spectacular articles, and I'm really looking forward to reading that as well. But there's many things that we don't know. Maybe going through all of these slides that I've just covered, you think we know a lot about fungi, but really these are just sort of cherry-picked examples from our body of knowledge that illustrate how spectacular fungi are, how strange they are. But most of what fungi do, we don't know yet. We don't know how much fungal diversity there is on planet Earth. Estimates of their diversity, species level diversity, range widely from hundreds of thousands of species, which is, in my opinion, clearly too low, to tens of millions of species. And whether or not that's too high, I can't say. But that's such a large range of estimates that it basically means we don't know anything about how diverse they are on planet Earth. And that scales down too. We don't know how many fungi there are in California. We don't know how many fungi there are in North America. There's only a few places on Earth where fungi are well enough studied that estimates of diversity are even close to being realistic. So, that's supposed to say identify. We don't know how to identify them. So it's really difficult for us to recognize most fungi on planet Earth. In some places, even well-studied places like Europe or California, there are still fungi being described every year. There's still identification challenges that are non-trivial, that we can't just guarantee that we'll be able to recognize a given fungus pulled out of its environment. We don't know anything about distributions of fungi. We have some maps for very small scales, usually state or country level around the world, but these are few and far between. We don't know sort of robust distributions of most fungal species on Earth, however many that might end up being. We know very little about their seasonality, their sort of phenology, their response to seasons that we've studied for so many years with plants, with birds. These are sort of well-known patterns. But with fungi, we have sort of rough estimates of seasonality. We talk about fall fungi and winter fungi and spring fungi, but what they're doing in the off seasons, we don't really know. What it means to be a fall fungus, what controls that fall sort of character, their phenology, we don't know very well. Is it daylight? Is it metabolic relationship with their hosts? Is it something about soil temperature or a combination of all of those things? We sort of have a guesswork at some of these, but we don't have real robust pictures of what's going on there. And then last but not least, perhaps population trends. We can't begin to say whether or not fungal populations are growing, are stable, are endangered, are invasive. There's a few fungi that we know are invasive, but beyond that, it's very difficult to sort of estimate how many individuals are around for any given species, and thus it's really difficult to estimate a population trajectory. We don't know how to conserve fungi because we don't know what their populations are doing. And this is sort of the most challenging aspect of fungal biology that could be resolved in the near future to make conservation more effective, more data-driven. But we're a long way from it. It's very difficult to monitor populations of organisms whose bodies are always underground, always out of sight, not really amenable to human perception. California is where I thought I would focus my attention to start to answer some of these questions, but it turns out that was a far too large of a scale. And so now I focus on Santa Cruz County, which conveniently is the second smallest county in the state. I can drive around it twice in a day if I feel like it. And it's probably just about right as a manageable, bite-sized piece of land to learn comprehensively the fungi that live there. So that's where I've been focusing my attention. And this is the part that starts to get into citizen science, or what I'll refer to later as community science, and I'll talk later about what the difference between those terms might be. But in Santa Cruz, I'm not studying fungi on my own by myself. I'm using iNaturalist as the primary tool to gather observations from other people, to see what other people are seeing, to see what other humans are experiencing in the woods, and to try and communicate with them about those experiences and ask them what patterns they're observing, what fungi they're finding. And for those of you who haven't used iNaturalist yet, there's a lot of resources about iNaturalist that you can find online. I'm not going to cover it as much in detail in this talk as I do in some of my talks. But basically, it's a citizen science platform. It's a database that has a website associated with it, as well as an app that you can download for your phone, your smartphone. And you can submit observations of living creatures from anywhere on planet Earth, any kind of organism, not just fungi. It's going to be birds, plants, insects, marine invertebrates, whatever you can find and observe, you can upload to iNaturalist, and a community of people will help you identify it. So what does this look like? This is a little map of Santa Cruz County, and it's a map of all of my observations of every living organism, all kinds of living organisms, not just fungi. The pink dots here are fungi, the green ones are plants, the purple ones I think are animals, and the blue ones might be, I don't know, fish. I can't remember what those represent. But you can see that there's big parts of my home county, even this county where I've focused on, that I haven't been to, that I haven't surveyed very well. And for those places, to learn about what organisms live in those places, I need the help of other people. And these other people can't always be professionals for a lot of reasons. I don't have access to professionals' time, and professionals don't necessarily have access to those areas. And what I mean by professionals is professional scientists, professional taxonomists, people who are trained in some way to observe organisms, to identify organisms, biologists. Most of these observations that I get from people are from people like you and me who are not PhDs, not classically trained. We're just biodiversity lovers. We're enthusiasts about living creatures. So by making a project like this in iNaturalist, where I can involve other people, where I can pay attention to what other people are seeing, where I can communicate with other lovers of nature, I've managed to gather something like 30,000 observations in just 4 or 5 years, which is an astonishing number of observations. That is not something I could ever have approximated by myself. They've recorded something like 1,127 species just of macrofungi in Santa Cruz County in those 5 years, which shows that even if not every species that they recorded is exactly accurate, the order of magnitude, the power of observation of people who are simply out and about because they like being in the woods is really strong. It's a really amazing tool to leverage this citizen science platform. So this idea that anyone can submit to this allows us to get these very fine-grained, long-term pictures of living creatures around us. And just to point out, this is 1,834 people. That's almost 2,000 people that have contributed to this project. Most of them I have not met. I've never trained them. I've not given them any formal rules for how to participate, for how to observe. They are going out in the woods, doing whatever it is they're doing. That could be walking their dog. That could be looking for plants. That could be looking for birds. And they just happen to make an observation of a mushroom, which happens to be my area of interest. And in doing that, in taking that extra moment taking that extra effort to share what they've seen, I get this extremely valuable data from my own project, for my own research, and for our general knowledge building about biodiversity in California. And this is something I want to talk about later as a way of creating knowledge by accident, not training people, not being formal, not being official, but simply letting people do what they love to do, letting them be at play in the woods, letting their brains wander around the environment and pay attention to what they want to pay attention to, and simply thanking them for it and sharing in that perception of the world and doing something with it, taking their observations, their shared data, and finding out what stories we can extract from it, what we can tell each other about the living things around us. So when you scale up from one person, this is what my map looks like, to the microflora of Santa Cruz County, which involves almost 2,000 people, to then the state level, we've now got something like 17,000 people participating over the course of the approximately 8 years, I think, that this project has been aggregating observations, maybe more like 10. But in that time, 17,000 people have contributed, recording something like 2,500 species of fungi. And astonishingly, I don't know how out of date this data is, it might be quite out of date, but 234,127 observations, 250,000 observations in that time is, I don't know what to compare it to, but for me, this is really heartening. This is something that gives me hope for our ability to sort of stay in touch with what's happening in the natural world as it changes rapidly these days, and to sort of attempt to respond to it in real time. If we can detect organisms this efficiently, we have a chance of finding out when they're in trouble, finding out when they might need our help from a conservation perspective. And you can see that in the, I think iNaturalist was publicly available first in 2008. People have just participated more every year. Every year there has been greater interest, greater input, greater participation from people around the world. It is an exponential growth phase right now. This is, I think, their last year's statistics for all observations of all organisms around the world. So you can see there's really no slowing down in iNaturalist participation at the moment. People are excited about this. People use it, not just in California, not just in the United States, but Mexico, India, New Zealand. These are all major centers of interest and participation in iNaturalist. So it has global appeal, and it's growing. There are people finally starting to catch on in Russia, which was for a long time a big dark spot in the iNaturalist observation map. But it no longer is. People seem to be catching on finally there. So I'm really excited as to what this data looks like 10 or 20 or 50 years down the road, when we have just an incredibly deep picture of what organisms are living around us on planet Earth, what they're doing, where they are, how they respond to seasonality, how their populations might be changing. All of this can be facilitated through the use of tools like iNaturalist. And we normally think about it as a citizen science tool. And I just want to tell you this little story of how new and different our ability is to detect organisms these days. So this is an organism called Scomida Fimbriata. It's a little purple mushroom, just the size of your thumb. And it smells like grape soda. It smells like candy. And I've never seen it. I would love to see it. I would drop everything, get on a plane, and fly to it if someone could guarantee me that I would find this on the other end. I did not see this, but my friend Ryan found it. He was in Alaska last summer at the end of the year when they were having some big wildfires up there. He was there for an ornithology conference. And after the conference ended, I think he went out for a long hike somewhere on the Kenai Peninsula near Seward and found these mushrooms, didn't know what they were. He's not a mushroom taxonomist. He's a birder, but he was texting out these pictures to a WhatsApp group that my friends and I have. And I was sort of absentmindedly scrolling through it, not really paying attention. Most of the mushrooms he posted were common, but this one caught my eye. And my jaw sort of dropped, because this is a pretty much a mythical mushroom. It's extraordinarily rare everywhere in the world. I think it had only been seen twice before by anyone ever, anywhere, both in Europe. And so to find this in North America was unexpected, and I was pretty jealous. And And I told him, hey, can you go back and get this mushroom? And he said it was 5 or 6 miles down the trail, 10-mile round trip. But much to my appreciation, he went back and he got them for me. And he took some notes on them. He dried them out on his dashboard of his rental car, took them back to the airport in Anchorage, flew back to Los Angeles, and sent me these dried mushrooms. A month later, I had them in my hands. I sent them out for DNA sequencing, and we had confirmation within another month that they were pretty much 100% match to the type specimens of Scomida Fimbriata from Northern Europe. So you see that there's a technology like WhatsApp, which is really just a social media platform that's very distributed, very sort of nimble, that friends can use to sort of spam each other with what they're seeing, share their observations with one another. And no one was trying to do science that day. There was no goal, there was no hypothesis, there was no survey method. But by sort of sharing with each other in real time what we're seeing, these extraordinarily rare organisms that you might only come across once or twice in a lifetime don't escape our sort of perception. We can stop and sort of capture the fact that they were there on that day and have a record of their existence and sort of be more attuned to their place on planet Earth and what they might be doing. This was a story that I never could have believed came together, and when it did, it really galvanized my belief that tools like iNaturalist are the way forward for biodiversity study. It's not professionals who have the keys to sort of understanding biodiversity in the future. It's amateurs, people who simply love being out there and sharing their observations with one another. So what does this show? It shows that amateurs can observe, record, and share in real time, persistently, meaning year after year, over the entire globe. And we're getting better at it every year. That is no longer in doubt. I don't think anyone can question this anymore. But where do we go from there? And what is the science part of citizen science? At what point does this become hypothesis testing? Or does it? Maybe it never does. And I'm not asking these questions rhetorically as if I have the answer. I'm asking these questions because I legitimately wonder, and you are going to be part of the community that wonders with me and answers. Not necessarily answering by coming up with answers, but by simply doing what you do. We're going to find out what this community does simply by doing it. So what can academics do to go back to professional scientists? What are they going to do with this new kind of data that we're generating that wasn't from any sort of protocol, that wasn't any sort of survey method, that had no hypothesis behind it? I think academics can start to engage the data. They can start to take what we're providing and ask new kinds of questions about it. Maybe questions that are more like stories, maybe questions that are more like narratives. And I think most importantly, before they start doing any of this, they can become participants themselves. They can start doing what we do as community scientists and simply observing, simply getting back to basics of being out in the wilds and observing what other organisms on planet Earth are doing. Kenichi Ueda, who is one of the co-founders of iNaturalist, says that one of the most important steps for academics who do research using iNaturalist data is to start by simply being familiar with the platform, how it's used, what its limitations are, how the community behaves, and that is best achieved by simply being part of the community. To give you a sense of why I'm not totally satisfied with iNaturalist and why I'm moving away from simply talking about citizen science in general and instead talking about community science, I want to illustrate what this meadow looks like on iNaturalist. And this is a meadow that is just to the west of the UC Santa Cruz campus, where I went to undergraduate and where I still advise students and teach classes. And this meadow is called the Meema Meadow. It's a really famous meadow. I don't know if you can tell from the map here, but out in the middle of this big expanse of annual grasses and wildflowers, there are these little sort of pale pockmarks. And there are these hills that are interspersed among sort of wet depressions. And the alternating pattern, this sort of checkerboard of dry hummocks and wetter areas between them, makes for an extremely diverse assemblage of wildflowers and just wildlife in general. There's a beetle called the Ohlone tiger beetle. It's metallic green, it's fairly large, and they're very rare. They live only in sandy areas like this in Santa Cruz County. They're endemic and they're actually federally endangered. So it's a really special place to be for biodiversity in general. But this is how you experience it on iNaturalist. It is a map with a bunch of dots with a bunch of observations that, although they might be illustrated with beautiful photos in some cases, don't feel like my experience of the meadow itself. This is not what I think of when I think of the Meema Meadow. What I think of is maybe a landscape like this, sort of a memory I have of driving up past the campus on a rainy, foggy day when I was supposed to be going to class, and instead of going to class, I got off the bus and ran across the street and rolled around in the muck in this meadow instead, and just sort of delighted in the fact that it was foggy and wet, and just sort of I knew what wonders were coming in the spring because the soil was getting the water that it needed, that it wanted. This is what I think of when I think of the Meema Meadow. I don't think of data. I don't think of observations. I don't think of any hypothesis. I just think of my love for the place itself. And I tried to sort of sum this up in a little collage I made of the organisms that live out there. And these are all organisms I'd seen at the Meema Meadow. Garter snakes and diconicas and lupins and soaproots and brodeas and red-tailed hawks, and down at the bottom there is a tiger beetle. And this is sort of what I think of when I dream about the Meema Meadow, or when I wake up in the spring and I know that I'm going there later in the day. This is sort of the experience I hope that my heart will have, something like this. And these are the visions that keep me coming back to natural history. It's not, I don't dream of spreadsheets. I dream of this. And I think that's what most of us are doing when we go out and take pictures of organisms. And that doesn't come without its costs. And I want to talk about that in a little bit. It feels very innocent or wholehearted to sort of just wallow in this beauty, but I think none of you are confused about the fact that this beauty is in danger in some places. It's not guaranteed to be there forever, especially if we keep behaving the way we are as a species. And we'll talk about that more in just a little bit. But community science as an alternative way of referring to citizen science has gotten, I've become more convinced by it. And some of the reasons for that are that citizen as a word took on sort of a negative connotation, especially in the past 4 years, where there was a lot of tribalism amongst Americans about who was a citizen and who wasn't. And in that respect, I think some people were turned off by the idea that citizen science was something done by citizens. And maybe that's a trivial concern, because I think many citizen scientists would immediately tell you that's not what they meant. But nevertheless, the word has its meaning, and the word has its flavor, and it strikes people the way that it does. So I began to ask myself how community science might be more appropriate. And in this sense, I like it better because it seems to lower the barrier to entry, which is what iNaturalist is all about, and it's what my approach to citizen science is all about. It's allowing anyone to participate, regardless of what they know how to do already, what they ever want to do, what they refuse to do. If they don't want to participate in a way that we understand, that's fine. People's neurodiversity is actually what makes this database as rich as it is. And I'll show you some examples. A friend of mine who is particularly attuned to slime molds is a good example of this. But community science also seems to make room for play, which is, I think, what most of us want to do. Instead of going out and laying out quadrats or taking transects or counting to very high numbers in very concrete, rigorous ways, I think most of us want to go to the woods and play. And that is not something that precludes your participation in science. I think science needs to figure out a way to incorporate that into its modality. And ultimately what this results in is knowledge creation by accident, which is to say we're building libraries of understanding, huge volumes of understanding without trying to, but simply by experiencing and talking to one another. You can't help but create knowledge. And fundamentally, there is no destination. There is no end goal to science. And I think that is something that I appreciate more when I think about the community doing it rather than the science itself. These people don't want to stop doing it. There is no end point at which I could say, okay, we know enough about biodiversity, you can all stop now. There is no amount of data that I would ever be satisfied by. I want the organisms themselves. I want the experience itself. And this is a picture of some of my students in the past couple of years. And I think when I look at these photos, I can see that this is true when I look at their faces and when I look at what they're doing. This guy is not taking pictures of those mushrooms because he particularly has any idea of how it will factor into someone's data scheme in the future. He's doing it because he finds this little conjunction of leptonias and waxy caps utterly beautiful and almost inconceivable that it's something that surrounds us in the woods that we are all a part of, all have access to. So this is what I try and cultivate when I take people out on my classes, is to simply stop as often as possible and just be present with the organism at hand and realize that there's no guarantee that we would share our time on Earth with it. And nor will we necessarily have a guarantee that they will be around forever in the future. That middle photo there is a student of mine named Julian looking up the trunk of a particularly large California nutmeg tree that in all likelihood burned in the CZU fire this past August. So that tree is probably not there anymore. That whole grove may not be there anymore. And I'll talk more about the CZU fire and the California fires in general in a few slides here. But this is what I see when I see people out. You know, the smartphones are in these pictures. People have their smartphones at hand, but I don't think it's because they're enraptured with the smartphone. And I don't necessarily think the smartphone is getting in the way of being present with that organism. I think it's a tool that people use to sort of make a concrete, sort of a keepsake, a way of cherishing that moment in the long term. They go back to that day and remember it more viscerally if they have a little a picture or a sound recording or a video to remember it by. So when I say the data is not the destination, it leaves the question of what is the destination, or what are we doing? And I think for me, and I don't know if this is the answer for everyone, but I suspect it's the answer for some of you, we're going out to delight in the aesthetics of the natural world, to see what there is to see, and to be astonished by it. Simply to allow our fascination to take hold of us and to guide our steps through the woods in any given day, or the tide pools, or if you're going snorkeling or scuba diving, whatever the environment that you're in, is to simply let your fascination move you along the landscape, and simply to experience wonder. It's not really that mysterious why people are out there. We all feel this wonder in the woods, and that's, I think, what we're after. This is a picture taken by my friend Allison, who I was telling you about, who is really interested in slime molds, but she took it to an extreme really rapidly that I've never really experienced before. She started doing macro photography at a level that I don't think many people do. I think she's recently been featured in a lot of, I think she was in National Geographic in their German article on slime molds this year. But it's difficult to take a picture like this and show it to someone and ask them what they're seeing and get the right answer. Most people simply have no idea what they're looking at. And yet these things are all around us in the woods every year. And Allison notices them, and she takes these pictures and brings them to people's attention in a way that I think draws those people closer to the woods, draws people more into touch with the organisms around them, makes them seek out these organisms they might not otherwise have thought about. So I really appreciate people like Allison, who's, for whatever reason, her brain gravitated towards slime molds and she took it to that extreme right away. We need more people like that. And I hope that as iNaturalist and community science becomes more and more popular, we'll experience more people who find what it is that lights their brains up and just do it as hard as possible and bring it to the rest of us so we can delight in it with them. And this is sort of an out there idea that I have, but it has become more and more pronounced the more I read about older naturalists. There is a chain of sensation. Your sensation, your five senses are yours to spend. Your gift is your attention, so to speak. And this has been the case for all of humankind. Naturalists have been paying attention and telling about it for years. And our goal is to learn from past naturalists, find out what they sensed, compare it to our sensations, and give some of that to the future as well. If you read accounts of past naturalists in California, you'll learn about things that are no longer here, that you wish were still here, but you'll also learn about things that you experience as well, that are still here, and that will feel richer to you because you know that other people have felt them in the past, and that they're part of these cycles that we can still observe. Mary Oliver has a poem that pretty much perfectly encapsulates this. Instructions for living a life: pay attention, be astonished, and tell about it. And if there was ever a poem that encapsulates, I think, what community science is about. It's this one. It's difficult for me to believe that things like this occur in the woods. This is two waxy caps that live in the deep redwood forests of Big Basin State Park near my home in Santa Cruz. And things like this keep me coming back to the woods forever because they're so unbelievably beautiful. You wouldn't necessarily expect that this would just be something you could go out and access in the woods. But there are moths like this. Everywhere you turn, you will find an organism that seems unbelievable until you've seen it and makes you feel that something deeper is going on in the woods than simply rote numbers at play. Nudibranchs like this keep me coming back to the tide pools. Birds like this keep me going back to the rainforests. Fish like this keep me snorkeling. All of these things seem almost too beautiful to be real, and yet there they are for us to delight in. But the Anthropocene, which is a term some of you may know, is this age of humans and their effect on the environment, and potentially putting all of this beauty at risk, or at least degrading it, making it more difficult for organisms like this to make a living around us. Some of the sort of key patterns of the Anthropocene are mass extinctions, defallination, which is a word that has become more popular recently as a way of referring to simply the declines in populations of organisms up to the point of extinction, but not including it. So organisms that we don't manage to totally extinguish, but we still reduce the numbers of their individuals to a point that they may be reduced to just a fraction of what they used to be in our lifetime in one human generation. As well as homogenization. So this is organisms becoming more like one another across the globe through invasive species, through homogenization of habitats, through extensive agriculture. And these are all things that are negative in my estimation. These are things that make biodiversity less rich, that make organisms less able to live alongside human beings, that make us more impoverished because we don't have that biodiversity around us. If you look at results from ocean sciences, you won't find comforting news. 70–90% of coral reefs around the world are predicted to be bleached, which means to lose their zooxanthellae, their symbiotic photosynthetic organisms, in the next 20 years because of global warming. They're very sensitive to increases in water temperature. So this is extremely bad news for people who are coral reef biologists. If you haven't seen a coral reef, you might want to do it soon because it's not looking good for them in the very short term. Same goes for birds. Our recent data about birds suggests that defallination has hit them really hard. You know, they haven't necessarily gone extinct, or you know, there aren't a lot of examples of birds going extinct in the United States, other than the well-known examples of ivory-billed woodpecker, Carolina parakeet, things like that. But defallination has taken a major toll on their populations. So even if they haven't gone extinct, we have a lot less of them. The air is quieter. You know, Rachel Carson's idea of the silent spring seems more and more prescient as time goes on. And then I think most strikingly, or perhaps most frighteningly, insects, invertebrates. They don't get as much attention because they tend to be less charismatic in most people's ranking of organisms they're moved by, but they are down deep in the functioning of ecology. This is how the world turns, is the actions of organisms like beetles, like mites, like arthropods that, you know, make soil out of fallen plant litter. And when you see big declines in the diversity and numbers of invertebrates, you know that something really, really serious is going on and that we're potentially in for a rocky road ahead if we don't get a handle on it. Fungi are not exempt from this. Kabir PA and a bunch of associated researchers, including an associate of mine, Clara Chin, published this in early 2020. They estimated that the fungal diversity associated with pines in North America is likely to be reduced by something like 25% in the coming decades, probably due directly to climate change effects. So across the board, things are looking grim for biodiversity. And at some point, I started asking myself, why am I encouraging students to fall in love with biodiversity when it's really signing up for front row seats at a cataclysm, at pain? to experience this sort of sense of loss. And the answer to that question is still elusive for me, but something that it has forced me to think about is what community science does. What we do with sort of this turbulent emotion that we get falling in love with biodiversity and experiencing its the various threats that it faces in yearly scales, in decadal scales. And it leaves me feeling like this thunderhead inside, these flashes of pain, these pangs of emotion, but somehow this overarching beauty behind it that keeps me coming back to it, that keeps me convinced that I should be doing it and not something else. Just this year, in August, that sort of unusual, unexpected freak lightning storm that came up the coast of California and sparked the CZU fire in my home county of Santa Cruz, caused sort of unprecedented levels of fire across the state. That was sort of the beginning of the 2020 firestorm that California experienced. And this is a picture from Waddell Creek. The day after the CZU fire started, I drove up the coast and took this picture of the Monterey pine forest right before I left town, right before the air got too bad to breathe and I felt like I needed to leave. And these trees are all gone now. This was as the fire was bearing down on them from uphill, creeping its way towards the ocean. And I'm going to miss these particular pines because there's not many of them. These are Monterey pines. They're relatively rare. There's only 3 native stands of them left, even though they're a widely planted horticultural tree. And all of the fungi that were associated with these trees are likely gone up in smoke with them. Thankfully, these are likely to regenerate. They're really good at regenerating in the wake of fires. Monterey pines are sort of adapted for that. But that's not the case with all of the plants that were burned in the CZU fire. So if you take the iNaturalist map of the CZU fire, there were about 9,000 records of fungi that were made in the 10 years leading up to the 2020 fires within the area that was burned this year, just in Santa Cruz County. And finding this out, I find myself extremely grateful that all the people who were involved in making these observations did what they did in those 10 years. All of that data was sort of our last chance to get to know what those organisms looked like in this pre-fire habitat. And everything from here on out is going to be post-fire. All of that habitat is never going to be the same for a number of reasons. Not just the fire, but also for the overarching effects of climate change that we're already having the impacts on these forests, the drought and the increased or decreased summer fog regime. So recently I was able to get back into Big Basin for the first time since the fires, and this is what it looks like in there right now. This is a photo I took of redwood regeneration and sort of the ash layer in Big Basin State Park. This is right in the core of the park in the deepest, oldest growth redwoods. And unfortunately, a lot of those trees were burned very badly. It burned really hot because of the heavy fuel loads in this area. But they are regenerating. Redwoods are good at resprouting, but not all of the trees were. Probably every Douglas fir that was burned was killed. They're really fire intolerant in that sort of habitat. So we're going to see what it looks like. And fungi were already fruiting there. The cut end and burned end of this redwood, that white blob on the right there, that's a polypore taking advantage of these newly downed trees and starting to decay that wood back into nutrients that other organisms will use to begin to regenerate these forests. So fungi already have their role to play and they're already finding it. So I feel very proud of the organisms that I'm most attached to. They're doing their work there. But these questions of loss in the Anthropocene, of biodiversity under threat, and people who are in love with biodiversity the way I am, makes me think that there's this unaddressed need in community science, which is figuring out how to help each other deal with loss, how to help each other gain perspective on threatened biodiversity and not be constantly aggrieved by it or constantly anxious about it or constantly mourning it. And some of the things that I've found that help me deal with this are thinking about biodiversity as a nucleus for community. These organisms are sort of a focal point, a center point that I have met people that are now dear friends to me, people I've spent years and years with. And we met each other because we were all interested in biodiversity. And that's something that makes me feel less pained about it. This will continue to be the case for the foreseeable future. We always can meet one another over the table that biodiversity provides. And beauty is always a balm. You can always look at these organisms and forget for a moment about anything else that was bothering you that day, any sort of larger threat that they might have experienced, and simply appreciate them in that moment as gorgeous organisms, and let your brain take that in and be bathed in it. You can experience gratitude for them, that you still get to experience what it is that you get to experience. And then lastly, something that I have really felt a lot of comfort in is simply experiencing or acknowledging that this is not the end of the line for biodiversity. Nature bats last is a common phrase, but nature always wins is a perhaps less appreciated truism. And that doesn't necessarily mean what you think it means. I don't mean nature doesn't suffer greatly at the hands of what people do to it, but this is ecology fundamentally. What we're experiencing right now, climate change, fires, drought, that is what ecology looks like. It's the consequences for the actions of a species. And as humans, we're having consequences come home to roost for us. We've behaved in a certain way for a century now, and there's simply no escaping the ecological ramifications of it. You know, we will lose biodiversity, but biodiversity will always bounce back, even if it's not the biodiversity that we knew and loved and cherished. So the perspective sometimes helps, knowing that organisms of one sort or another will be there long after we have had our most pronounced effects on the climate. Things will get cool again. Things will probably get hotter again after that. And even if it's not the organisms that I knew and loved, there will be organisms around for us to appreciate. Like whales. This is one that I saw off of Moss Landing, very near shore. I was standing on the beach when I took this photo. And encounters like this remind me that there will always be something there to astonish us. Always be something there to take joy in, even if it's just for a moment. Gary Snyder wrote this poem that I think is becoming more and more important in my natural history practice as the years go on. But if Mary Oliver gave us some very brief instructions for how to do community science, Gary Snyder gave us excellent instructions for natural history living. Stay together, learn the flowers, and go light, which is what I try and do when I teach my classes now at UC Santa Cruz. We're focused on the organisms. They are the table that we're meeting across. We're all there to pay attention to fungi and plants and whatever else we might encounter while we're out in the woods. But I want the students to know that equally as important are their connections to one another, and their connections to future humans and their connections to past humans. And that's really what I've started to take as core to my natural history practice, the community aspect of community science and not so much the data. So thanks to the HBMS for having me here tonight, and I hope that we can all get back to doing this in the nearer future than the later. I look forward to seeing you in the woods.
That was great. Since nobody else can tell you, that was great. Somebody on YouTube actually did tell you that this is a great talk.
I'm glad to hear it.
We did have one question come up from Barbara Ching. She asked, is there a bright line? Okay, do you want to answer it?
Oh, wait, sorry. No, I just saw her name come up. I didn't see the question itself.
Okay, so the question is there a bright line or a clear boundary separating a detailed observation from the scientific method? Does phenology, for example, which sequences the unfolding of spring or fall, unfold apart from science?
I think I can probably give a blanket answer that in my mind, there are no clear lines about anything ever. So I certainly could not immediately guess at one. I don't think there's anything that immediately separates an observation into more the casual observational side of things or the more scientific side of things. Obviously, the more data that is included, whether that's a picture, a date, a location, a time, microscopy, DNA sequences, the more you add, the more likely it is to be usable in different paradigms of science. But that doesn't mean that a very casual observation can't play its own part in a bigger picture about what organisms are doing. There's a lot of casual observations that are really valuable for whatever reason, just as a record of an organism being in a place on a day. Sometimes that just shapes future efforts. You know, even if you can't confirm it from the observation itself, it suggests to someone else, hey, this is the place to go look for this thing. place you might encounter this organism. I don't know if that answers Barbara's question.
Okay, well, you also got one more question, and then we can get
Okay.
some feedback from Barbara if she wants to. So
Sure.
Shannon asked, one of the goals for an Inapp project on mushrooms that you spoke of earlier in your presentation, you referred to sequencing. I wonder how you would have been able to find the funds to cover the costs.
That's an awesome question. So DNA sequencing is one of the sort of most accessible immediate steps that mycological societies can take to empower their communities to make really high quality scientific observations, things that like actually generate amongst the most valuable new pieces of information. And it's not as expensive as you might think. Costs are coming down. There's more resources for getting DNA sequences than there ever have been. This is the time to be doing this as a community. You can look at the North American Mycoflora Project recently changed names. It's now Fundus, Fungal Diversity Survey. But if you go to their website, you can get in touch with a bunch of resources for how to get grants to do that kind of thing, and how to use iNaturalist to sort of structure club-based projects to do DNA sequencing of biodiversity.
I see a question in the YouTube. I got to jump around to my windows.
Okay, while you're doing
so
that,
somebody
acknowledge Richard Linus here and say that he wants to personally acknowledge the role of serendipity in his practice. I am looking for more disciplined ways to find serendipity in the world and in the woods and in my life, and I appreciate that comment, Richard. Richard is from the Fungus Federation in Santa Cruz, so I know him well, and I'm glad to see him here. Yeah, serendipity is always fun. There's so many stories of serendipity and finding mushrooms on the same day as someone else, or seeing a mushroom that you always wanted to see on your birthday or something like that. They seem to give you little clues that you're doing the right thing that way. Okay, so back to this YouTube question you were reading.
Okay, so you had some people who were pretty excited about your story about the meadow and connecting with community and passion in science. And then somebody asked if you had any interesting discoveries or curious finds since the publication of Mushrooms of the Redwood Coast.
Oh God, lots, yeah. I mean, the publication of that book just basically meant it's when we concretized its obsoleteness, right? Because then it's just sort of a summary of what we knew at that moment, soon to be outdated, which is the goal, is to have that be a rung in the ladder that is soon superseded and someone else makes something better. But one of, I think, probably the most sort of galvanizing experiences with relation to that is we included a fungus in there called Dendroclobia pycnoramella, which is a mushroom that isn't described yet formally. And it's super, super, super rare, incredibly rare, even rarer than one that I mentioned, the Scomida that I mentioned that Ryan found. And we included it in the book, sort of, it was a bit of a gamble because, you know, it wasn't formally described. So we were including it sort of against some judgment. But we knew that we had the best chances of finding it again if we could get other people looking for it. If we could get someone else paying attention and sort of knowing that what they found was special by having it seen or having seen it in a book somewhere. And I thought it would take a decade or more for someone else to find this mushroom. But I think it was within 1 year, someone found it in Southern Oregon. So it really paid off. Our decision to include this super rare, poorly known mushroom in the book was actually the best way to get more information about it because it roped in other people. It sort of took advantage of that community of people and all their eyes and all their ears and all of their sensation on the land they covered, got us more information about that species sooner than later.
And then we have another question in the Q&A. Is there really any value in entering common fungi into iNaturalist, since there will already be plenty of sightings already listed there?
I cannot overstate how important it is to add observations of common organisms. They are almost what I want more than anything. Obviously, it's really useful to get and really fun to get observations of rare things and beautiful things, but the less appreciated mushrooms that are not aesthetically as striking or that are just really common and we feel like we see them all the time, that's not always the case. You know, things change. Sooty shearwaters are a really common bird, or used to be a really common bird in the Monterey Bay Area. And they're still really common. You go out there and you'll see 100,000 in an afternoon, which feels like, okay, if you can see 100,000 birds in an afternoon, it must be a common bird, right? But the story that is obscured when you see those is that, 50 years ago, you would see 1 million instead. So it represents a 90% loss in their numbers in just a few decades. So just because something is common now doesn't mean that it will always be common. And just because the numbers are high, it doesn't mean that there wasn't a huge reduction over time. So observations of common things are still super important, not only for things about population, but about seasonality. The more data points that we get, the more we can actually say things about what a fall mushroom means. So what does it mean when we see a sort of a strong response pattern in the autumn months versus the spring months, and what sort of environmental variables were that shaped that? So yes, please don't ever stop observing common organisms. Sincerely, me.
Okay, and then you got another one in the YouTube, which is, what can we as community scientists do when it comes to finding observations to help biologists? How and what specifically should we detail in an observation?
So my immediate answer is to do what moves you, is to just go out there and do what you like doing, because that means you'll keep doing it longer. And that's mostly what I want, is for people to go out and experience organisms and talk to each other about them. Now, if I want to give a sort of a deeper answer, or like a more less philosophical answer and more practical answer for people who are actually looking to sort of generate some of this data, which I should point out, it's not that I don't value the data that is being generated, but it's not the end goal for me. It's not the be all end all. But if that is what you're after, habitat associations are something that we're sort of piecing together in a more rigorous way about fungi still. So if you can include observations that you make about what the habitat's like around the organisms that you're finding, that's always useful. What other organisms they're associated with, whether that's insects, invertebrates of some kind. I think there's quite a bit left to be done. I mean, if you get a microscope, which isn't as big of a financial or intellectual investment as it sounds like to some people, people often think that's like very advanced, but it's really not. You can actually get up to speed on doing microscopy pretty quickly. And most people find it really enriching, and they love doing it once they start doing it. That's another way you can add valuable data to your observations. But another way that's really easy is just to do more of them. The finer grained our maps are of where mushrooms live and when they do their thing, when they fruit. Both of those things give us sort of more power to talk about what they're doing, what their populations are doing over time.
I would also recommend that people smell mushrooms for their own personal enrichment.
Oh yeah, I'm like writing a whole blog post on smell right now, so I really appreciate that reminder, Maria. Smell is like, some of the smells you'll encounter in the mushroom world are really unexpected. There's like I said, grape soda for the Scomida that I mentioned earlier, but you'll get aniseed, you'll get phenol, you'll get all these things you sort of never would have expected to find in the woods. And that's just sort of a sense that we're not very good at using as Americans. We don't have a lot of good words for smells, and mushrooms are an awesome foothold into getting better at it, getting better at using one of your senses.
If you are just starting to smell mushrooms too, you have to tell yourself to ignore the smell of dirt first, and then you'll get there.
Yeah,
you got another question.
Okay, go for it. Go for it.
Are you concerned about mushrooms being misidentified in Inapp?
Yes, but not very much. The whole goal of citizen science is that it's very tolerant of that sort of noisy data. It's okay to make mistakes there. That's the whole point, is we're reducing the barrier to entry to allow people to just get in there and make mistakes, and we'll fix it later. And that's sort of the responsibility of the professional taxonomists, the people who are spending more time doing it in a serious way. That is what our value to the community is to help come back through and sweep through that data and give you an idea of what you've seen and correct those mistakes where they exist.
And then I guess this kind of goes along with that one. How do scientists deal with opportunistic observations that weren't collected in a systematic way through time and space?
By doing math that is beyond me. I'm not really good at statistics, but there are ways that you can sort of account for the fact that data was not systematically collected and make some adjustments that relate it to other organisms and intensity of sampling by other observers in that area in that time. So there are ways to handle it. You won't always be able to do every statistic that you wished you could, because like you said, it's not systematic, but that's okay. I don't think we always need that.
And then we have another one here. Should we hide our coordinates of edible fungi and or rare fungi when we post observations on Inapp?
That's a good question. So the idea here is that rare fungi might be sensitive to human impacts in some way, and that edible fungi obviously are sensitive to human impacts in the way that people go out and forage them. And whether or not we should sort of worry about hiding the coordinates, making it harder for people to find these things, that's a good thing to be concerned about. But in some cases, this is automatically done. So I have tried to make it so that iNaturalist coordinates for choice edible mushrooms are displayed only coarsely, so people aren't zooming in on your exact chanterelle spot. That's easy enough to do. And I try and make that sort of automatic so you don't have to think about doing it. That's something to think about more about how we use citizen science databases going forward. eBird has had some of these similar challenges, but I wouldn't be too concerned for rare fungi, because the chances that there's going to be a large community of people going out to look for your Neohygrophorus patch is low, to say the least.
That is the last of my backlog of questions. I don't know if any more are coming through. Somebody said, LOL.
I think that was Rebecca.
Okay.
Okay, well, thanks for having me, and I miss you all.
Yeah, we miss you too. Thanks for a wonderful talk.
It rained here for the first time this season today, so I'm excited to finally start getting into it myself.
Ooh, do you want to answer one more question? One more came through.
Of course. Oh, and I see CJ there. Hi, CJ.
Okay. Does the idea of a systematic mycoblitz from time to time hold over to you? You mentioned the meadow above UCSC. I think of forays were done at Quail Hollow. Soquel Demo Forest. I wonder where else.
I'm not really sure what that question meant, but the answer is yes. Quail Hollow, Marshall Field, Soquel Demo have all gotten lots of attention from the Fungus Federation over the years. Oh, Allison was in this talk. Hi, Allison. Cameo appearance. Hi, Luke.
Okay.
Right,
yeah, we had a great showing.
I'm gonna go eat
There's
chicken.
Tons of people on YouTube also.
Cool.
Okay.
Bye, everybody.
Hi, Christian. Thank you.